Good morning once again. It's time to start because it's 11 o'clock. Thank you for joining me again to this lecture. It's about neurohistology again, and last time we discussed the nerve fibers and the structure of the neurons was discussed in the last lectures, and this talk is about nerve endings. You will see that we have two major kinds of nerve endings. We have the receptors and the effectors. This is what I'm going to discuss with you next time. Today I will talk about the structure and the types of the synapse, and I will give you a classification on this. I will also tell you some basic statements about the electric synapse. The chemical synapse will be more detailed discussed. I will talk about the general structure, the function, and I will also explain you classifications based on the morphological and also on some functional aspects according to Gray. And you will see that at the end of the lecture, some remarks on the neurotransmitters, because there is such a topic in the list of the exam questions as well. Okay, nerve endings and the definition of the synapse is the first part. This slide is already familiar to you. I don't want to explain the whole story again, just to remind you that the stimulus comes from the environment. It will activate the receptors of the skin, and these receptors are found in nerve endings. This is what I will talk about tomorrow. Then the signal is picked up and transmitted through the peripheral process and then through the central process into the spinal cord if it's about a spinal ganglion. And here you can see that this nerve fiber ends and gives the signal further to this interneuron. And then this interneuron will then go on and with its axon terminate in the neighborhood of the or on the surface of the motor neuron, and again there is a connection here. And then the motor neuron will fire and then will go with its axon to the periphery and will activate the muscle. This activation goes to another tissue, to the muscle tissue, and here is a so-called effector found. This is discussed again tomorrow. Again, we have the autonomic nervous system, where we have the preganglionic fibers. Synapse takes place in the autonomic ganglion, and the postganglionic fiber will again innervate, for instance, internal organs, which is also given by effectors, the topic for tomorrow. But what I'm going to talk about today is the interneural synapse, so how nerve cells communicate with each other. And in this scheme already, we have multiple interneural synapses. And if I want to define what a synapse is, it is not easy. I opened several neuroscience books to find a nice definition, but this is hard. Most of the times it is defined as connections between cells, specialized connections between cells facilitating the signal transmission. Now, if it's about signal transmission between nerve cells, it's about interneural synapse. This is what we are going to talk about today. But as I mentioned earlier already, we have nerve cells which communicate with other types of cells, like with muscle tissue or with glandular tissue. This is found in the autonomic nervous system and also in the somatic nervous system, in the periphery, the effectors. This is discussed tomorrow. In the pictures down, you can see here some nerve cell bodies in the brainstem, and here you can see some nerve fibers which approach these neurons, and there you can see that there's a close relation, close so-called opposition between these cells. So most likely there is a synapse found between these cells, but one could check this out more precisely with the electron microscope. On the other side, we see a so-called motor end plate. This is what I will talk about tomorrow. This is a synapse between the neuron and the skeletal muscle. It's also a certain kind of synapse. So now you know what a synapse is. And now let's talk about the synapses. First, I would like to classify them, that you understand what kind of types synapses have. So first of all, we have the synapse, and that we can divide into the chemical synapse group, and we can also define an electric synapse group. Electric synapses are found in higher proportion, in higher number in primitive creatures, but this doesn't mean that in higher order animals, in mammals, or in humans, non-human primates, or in primates, you wouldn't find it. They are also found in the human nervous system, but we have more chemical synapses, or their significance is at least higher. Electric synapses may be called as a gap junction or nexus, and you have to know about the electric synapses that they allow direct transmission of electric signals between cells. So in this case, there is no chemical substance release taking place. No neurotransmitter release takes place, just electric currents will go through from one cell to another. And because of this reason, the signal transmission is permitted in both directions. So if two cells communicate with each other, they will bidirectionally eventually communicate with each other. How do electric synapses look like? Similar figures you can find in any histology books. You have two cell membranes closer to each other, and you have a distance between the cells, an intercellular space of 20 nanometers. But in some areas, this distance is significantly reduced to 3-4 nanometers. And in these areas, the cell membrane has specific proteins, and 6 of these proteins form a specific channel. The proteins are called connexins, and these connexins will then form a channel, and the other cell has similar connexin proteins too, and the two cells will therefore be connected through these channels with each other. Sorry, I forgot to translate this, but I will do that. And then you have here the connection, and the ions can pass through these channels, and they therefore will activate or influence the other cell's activity very, very fast without almost any synaptic delay. The signal transmission takes place. So this kind of connection is very important because cells may be synchronized with each other. If a synchronized electric activity is required for certain cells, then you will find similar connections. Similar connections you have studied already in the cardiac muscle tissue that synchronizes the action of the myocardium as well. Also there is connection between neurons and glial cells, and if they communicate with each other, then these kind of electric synapses will be responsible for that. You have a lecture. You had a lecture earlier about the retina, and you most probably heard about the photoreceptors there. Photoreceptors also have similar connections, and you can give an example. The photoreceptors, if it's about electric synapse. So we do have electric synapses, but we do not have so many. And if it's about electric synapses, one has to state that the chemical synapse is more important in the mammalian and also in the human central nervous system. So that therefore, in the remaining part of the lecture, we will discuss the chemical synapse, which has a greater significance. Here you can compare them in this scheme. So in this case, the signal transmission takes place with the help of chemical substances. And these chemical substances are released from the first cell, and then they will act on the second cell. These chemical substances which transmit the signal are called neurotransmitters most of the times. The signal transmission, because the neurotransmitter is released from one cell. The next cell has some receptors for that, and only those receptors can perceive the signal which carry the receptor. the signal transmission is unidirectional, so it can go in one direction only. This is an important difference if we compare it with the electric synapse. So this scheme is also known already. I think you could remember this. We have the cell body of the neurons, the dendrites, and you have the axon. And you see that I indicated here some synapses on the dendrites, some are found also on the cell body. And if this neuron produces an electric signal, it is able to give it further to another cell through this synapse, for instance, which activates or inhibits the next neuron. So these are the synapses in this older scheme of mind. Now, if we zoom into one such a connection, I try to explain you the general structure of the chemical synapse in a very simple scheme. So this is the nerve fiber that comes to the synapse. This is the end arbor or the terminal arborization of the axon, which usually enlarges a bit. This is not necessary, but usually there is an enlargement. And this enlargement is simply called presynaptic structure or terminal nobor button. With this term, you may also define it. Now, here some quiet energy-consuming processes will take place. Therefore, mitochondria you will find in the terminals most of the times also. And then the signal will spread to the next cell, which has there the postsynaptic structure. Again, one can find mitochondria in the neighborhood. Now, between the two structures, there is a certain distance. The synaptic cleft is quite variable in size. It is usually 50 or less than 50 nanometers. One can, of course, find different data in the books. It depends on the kind of the synapse also. But of course, you will always find wide ranges in the literature on that. But it's around 50 nanometers or less. Now, the cell membrane here, which belongs still to the presynaptic structure, is called presynaptic membrane. And with the actual microscope in this presynaptic membrane, you can find the density, which is made up mainly by proteins. It's called presynaptic membrane density. And next to this presynaptic membrane density, one has synaptic vesicles. And these synaptic vesicles are usually loaded by neurotransmitters, which are finally released into the synaptic cleft, but I will come back to this. And on the other side, we have the postsynaptic membrane. And again, there is a density on the postsynaptic membrane, which is made up by proteins again, and these proteins will be required in the signal transmission also. So this is how the general structure of the synapse looks like. And if we would like to understand the function, then I would like to go into some details on the synapse. So we did this yesterday, that the action potentials, or not like yesterday, but last time, that the action potentials travel on the axon. The depolarization wave runs through the cell membrane to the terminal, and it arrives there. Now, if the action potential arrived to the terminal, it will open some channels on the cell membrane. These are particularly so-called voltage-gated calcium channels, which then will allow that calcium ions will enter the terminal. And because this happens, the calcium has several effects, and one could give a talk alone about this. But just to make it short, the calcium has one effect, that it will release the synaptic vesicles from their bond. They are bound to this so-called synapsin molecules. And if we would like to release the neurotransmitter content of these vesicles, we have to free them from this synapsin connection. And this happens, and then the chemical synapse can release the substance. How does that work? This slide is about the chemical synapses detailed function. So once again, the action potential arrives, opens calcium channels, and the vesicle will be released from this synapsin connection. Now, the calcium has also different effects. One thing what you have to know to understand this is that in the presynaptic membrane density, we have a so-called syntaxin and SNAP25, SNAP25 proteins, and they are called together the snare protein complex. This is located in the presynaptic membrane density. And also, the vesicle, which contains the neurotransmitters, has in its membrane proteins, and they are sensitive for calcium influx. And you have the so-called synaptotagmin, neurexin, which are calcium sensors. And there is another protein called synaptobrevin. And now, if the calcium comes in, that activates the calcium sensors, which activates then the synaptobrevin. And after this activation, it can already dock to the presynaptic membrane density, because the syntaxin will then interact with the synaptobrevin molecules. And this is how the vesicle is then connected to the presynaptic membrane. And after this, very soon, the cell membrane here, the presynaptic membrane, may fuse with the membrane of the vesicle. And you can see that the content of the vesicle can be now emptied into the synaptic cleft, which is an exocytotic process. Now, if the synaptic cleft has the synaptic the neurotransmitter now, then it diffuses very fast to the postsynaptic structure, where we have receptors for that. In the postsynaptic membrane density, we have neurotransmitter receptors, and they bind to the surface of those receptors, and they are most of the times, not always, but many times, ion channel coupled receptors. And if this neurotransmitter is bound to the neurotransmitter receptor, then the neurotransmitter receptors will suffer conformational change, and therefore sodium, for example, here, but there are also other examples, the sodium channel opens, and then sodium ions may then enter the cell. This will interfere with the cell membrane, with the membrane potential, and then it may activate the cell. In this example, we have an excitatory effect. However, if other kinds of ion channels are activated, inhibitory effect may take place, but I will come back to this point later. And now you see that this neurotransmitter is bound to the receptor. But of course, if you would like to activate the postsynaptic cell, you did this already, and we don't want to keep it active forever. So this has to stop somewhere. Therefore, there are several mechanisms how this can be finished. And one option is that the synaptic cleft has certain enzymes which are able to split the neurotransmitter molecules, and they are broken down. And these breakdown products, or the neurotransmitter proper in another mechanism, is taken up by the glial cells. Sometimes the presynaptic neuron proper picks up the breakdown products or the neurotransmitter per se, and then it may get reactivated, or there is a certain reuptake sometimes also in the neuron taking place, but glial cells can remove the synaptic, it can clean the synaptic cleft, and they can remove the neurotransmitters from there. And also the cell membrane is then recycled, because this synaptic vesicle is covered by a clathrin coat, and this will then again internalized into the cell, and then this vesicle will then work as a synaptic vesicle again. It is reloaded later with neurotransmitter, and then a new cycle starts up again. So this is very, very simplified and briefly how it works. And I'm talking about this already for 8-9 minutes. You may ask the question, why do I have to know this? Because I will be a doctor, so nice, but how do I use this in the clinical practice? So on one hand, there are several toxins and different kinds of diseases where then this signal transmission is compromised or is somehow influenced. So this spider, the black widow, has a strong poison, the latrotoxin, that will activate the calcium channels. And because of this, an uncontrolled excitation and emission of neurotransmitters takes place, and this is a life-threatening situation. And after the release of a large amount of neurotransmitters, no neurotransmitters are left, and therefore a kind of palsy occurs also. So the latrotoxin works here. And also you will hear about a disease that is caused by a bacterium called Clostridium bacteria. And they may produce a toxin, which is the botulinum or botulinus toxin, which is then interacting with the snare protein complex. And if that doesn't work, then the exocytosis is disturbed. And one may get infected by this bacterium, or more likely by the toxin of this. And a disease may occur which is related to the problem of muscle movements and eye movements, and the laryngeal muscles don't move properly, and palsy may occur in several parts of the body. But we can use this also for medical purposes, because plastic surgeons may treat the patient's face by this toxin in order to reduce the signal transmission towards the muscles of facial expression. And this is how one can may change the look of these people a bit. People say that these persons after treatment look younger. I'm not sure about that, but for sure the muscles of facial expression for a while get paralyzed, and therefore they don't form deeper grooves and folds on the skin. Therefore it may have some effect. And also the tetanus toxin is another issue, which is also a bacterium-produced toxin. This bacterium might be present in the soil, for instance, and if you cut your finger when you are doing gardening or something, and these bacteria enter your skin, you may suffer a tetanus infection, which is interfering with the synaptobrevin. Again, serious muscle cramps may develop, and one may die on this. Luckily, we have an immunization against the tetanus infection. So if somebody suffers an injury, it's always important to look after if the tetanus immunization was given or not, or in that case we have to give it. Okay, so these diseases, or this spider also with its toxin will interfere with the synaptic function, and therefore I wanted to talk about this a little bit more detail today. Then we move on, and now let's talk about the synapses. And in the coming minutes, I would like to explain you the classification of the synapses, and I'm talking about the chemical synapse now only. In this scheme, this is a drawing where people try to summarize the most frequently found common types of chemical synapses. And one can find here the axodendritic synapse. So these terms comes from the name of the cell parts which meet each other. Let's have a look. So number one, here you can see that there is an axon coming, and the axon is connected to the dendrite, to the shaft of the dendrite. There is a connection here which allows the signal transmission. Some books will define as an independent entity. Some books say it's a type of the axodendritic synapse, that we have an axon again that comes again to a dendrite, but it does not meet with the dendritic tree proper, but it meets with the process of the dendrite, which is the dendritic spine. We did this earlier. if you remember, and the dendritic spine forms a dendritic spine synapse, and it's a specific type of the subtype of the axodendritic synapse. It's always excitatory. And then we also have another option, namely that the axon which arises here, this is the axon hillock, we did this earlier also, where the axon arises. Other axons may exactly terminate to the origin of the axon and may directly stimulate, for instance, the activity of the axon, and this is the so-called axonic synapse. Again, it's a quite common type that axons come to the nerve cell body and they terminate on the surface of the cell and on the soma, the cell body. That's why axosomatic synapses one can also find. They may be inhibitory, sometimes excitatory as well. And then we have also other types, which I do not demonstrate on this scheme, but one can find them more rarely in the brain. But I listed here these names, and I will give you some examples for those, but not for all of them. Somatosomatic synapses may occur if two neurons meet each other with their cell bodies. The somatodendritic synapses, if the cell body communicates with the dendrite, and the dendrites may also synapse with each other. Dendritic synapses also exist. And there is a so-called en passant synapse, which is characteristic, for instance, in the autonomic nervous system. I will come back to this next time. So these synapses are not illustrated in this figure, but I would like to show you some examples for these synapses in the coming slides. So once again, common types, axodendritic synapse. Here you have a dendrite with electron microscope, transmission electron microscopic image. And there you can see how an axon arrives, it ends. Those who have good vision can see that there are some little oval round structures in those. That's the synaptic vesicles. And here this dark patch represents the densities, the pre- and post-synaptic membrane densities, and this is the synapse here between the axon and the dendrite. That's why we call it axodendritic synapse. Accidentally, in the same image, one can observe another dendrite here, which forms synapses with this dendrite. So this is a rare type of synapse, but one can observe them here also. The dendrodendritic synapse is visible too. Okay, spine synapse. Again, just some pictures. The dendritic surface is not flat. It's very uneven. You have many dendritic spines on that. I showed you earlier this Golgi stained specimen with dendrites and dendritic spines. If one prepares a freeze fracture technique, and then also you can see here the dendritic shaft, and there you can see how the processes, the dendritic spines arise from that. And if we use the transmission electron microscope, this is the cross section of a dendritic spine. One can see that it has this kind of mushroom shape, and then it has a stalk and it has a wider head. And there you can see, if you check it all over around, so this is the dendritic spine, and if you check it, almost the entire surface of it is covered by axon terminals. Here is one, there is one, there is one, and so on. And each axon terminal, of course, not all of them are exactly hit, but most of the times you can recognize that there's also one, that there are synapses here. And these dendritic connections, so these spine synapses are of excitatory type. They are always excitatory. Axosomatic synapse again, and some examples for that. So once again, the nerve fiber comes and ends and terminates on the cell body. And if you go to the classes in 3 weeks, if I'm right, you will study the spinal cord histology, and you will see the spinal cord preparations with Bischoff's impregnation as well. When I gave you the talk about the imaging and studying the nervous system, the techniques in general, I mentioned this impregnation, that you have yellow cells and you have a black or very dark brown nerve fibers everywhere. Myelinated fibers are mainly marked, but the nerve fibers, neurokeratin, and is also visible. Now, if we zoom in into these cells, and if they are conventionally cut, one can observe that there are little oval or round black structures on the surface of the cell. And if we check those out, they correspond to synapses, and the entire cell is almost everywhere covered by similar nerve endings, and they form the axosomatic synapses. The head, Auerbach, and foot or end feet are they also called in the literature. Now, you can imagine that if this cell has these little synapses on its surface, the cell is relatively large in comparison to these little connections. So finally, if the entire cell is covered by similar synapses, several hundreds or even more than 2,000 synapses can be on the cell body, and even more on the dendrite. So one single neuron may even get more than 10,000 synapses, but we have specific kind of cells which have up to 100,000 synapses. So this is a really big number if we consider that these fibers, these synapses will carry signals from different sources to the cells. There's a very strong convergence of information to one single neuron by these kind of synapses. This is required for the decision making, for instance, and how then the neuron fires will determine the action, of course. An electron microscopic image from an axosomatic synapse. This is the cell body, the perikaryon, marked by P, and this is the terminal, the button, and there you can see the membrane densities. There is a certain cleft here, the synaptic cleft, and you can also see these little bubbles. They correspond to the synaptic vesicles loaded by neurotransmitters, and you also see here some scattered mitochondria. All right, we have some rare synapses, and I don't want to go into details on this slide, but you will get this when you get the lectures about the cerebellum, because this so-called glomerular synapse is found, for instance, in the cerebellum. And here you will see that not only two, but multiple neuronal structures will meet in a compound synapse. This is the glomerular synapse. It has a glial cell-formed wall or capsule, kind of. And then so-called mossy fibers will synapse with the granular cell dendrites. But this signal transmission is then influenced by additional nerve fiber endings which belong to the Golgi cell axon terminal. And that's inhibitory, and it will eventually inhibit the signal transmission between the blue mossy fiber and the red granular cell. About details on this and how this contributes to the cerebellar function, you will study more later. I just mentioned at that time, if we refer back, you know what we are talking about. So this is a specific kind of compound synapse. Also, another rare synapse type is the so-called ribbon synapse. This is found in the sensory organs. In this case, in this synapse, you have a band-like invagination of the cell membrane. And around this, many synaptic vesicles are found. And because it increases the surface where the release of the neurotransmitter can take place, it is characteristic for those cells where a really strong, fast, highly efficient exocytosis of neurotransmitter is required. And we also know that there is a certain baseline tonic activity. So some neurotransmitters are released continuously, but eventually, if it's needed, then like a blast, it's a very fast expansion, like exocytosis taking place here, which is then characteristic for the ribbon synapse. There are also specific subtypes of that, depending on how the neurotransmitter vesicles are distributed, but I don't want to go into those details. It's enough if you know that they are found in the sensory organs with electron microscope. You can find these kind of synapses. They are rare synapses. All right, and after this, I will go on now, and I will move to the to another classification, which is quite frequently seen in books, and this is something that we also would like to get back in the exam. So, if the question arises, what kind of interneural synapse classifications we have, you may give this morphological classification, what I did until now. So between what and what is the synapse found, and axosomatic, axodendritic, and so on. But if we check the function of the synapse, and if we check also the morphology that of course is interrelated with the function, then we come to another classification which was defined by Gray. And there is the type 1 synapse, and there's a type 2 synapse according to Gray. Because of the histological, electron microscopical histological features of these synapses, we may call them asymmetric and symmetric synapse, depending on which book you use. Some books will use the Gray 1 and Gray 2 terminology. Others will prefer to say asymmetric and symmetric synapse, but that's basically the same thing. So let's have a look. I indicated the most important point here, that asymmetric synapses are excitatory. So they stimulate the postsynaptic cell. In the type 2 symmetric synapse, the Gray 2 synapse, an inhibitory function takes place. So the postsynaptic membrane will be inhibited, will hyperpolarize, as you will see in a later slide of mine. So here some statements are listed which allow you a comparison between the two types of synapses. If you look at the picture down there, you see that we have round vesicles, synaptic vesicles here. Their diameter is around 48 nanometers. And here you can recognize easily the synaptic cleft. It's relatively wide. And you also see that the presynaptic membrane density and the postsynaptic membrane density are of different thickness. This is why we call it asymmetric synapse, by the way. And the postsynaptic membrane density is definitely thicker. They are found in dendritic shafts and in this and eventually in the dendritic spines, as I mentioned here. They should be excitatory. On the other hand, this synapse looks different, since both the pre and the postsynaptic densities are very tiny, almost hard to recognize, but they are there. Narrower synaptic cleft, you see, in comparison to that one. And also, you see that the vesicles are different. They are a little bit smaller and eventually oval in comparison, in contrast to round. So this helps you to distinguish the excitatory from the inhibitory synapse, which is this one at electron microscopic level. They are found on the cell bodies, but they do not occur on dendritic spines, because that was the excitatory one. So please compare the Gray 1 and Gray 2. And if the question arises, what are the differences, your examiner wants to get these points most likely. Right. And the final part of the lecture today is about the neurotransmitters. And there is a half topic, if you will check the list of the exam topics, classification of neurotransmitters or something like that. So point 1 is already that what we approximately discussed already, but let's have a look once again that we really get this. So once again, presynaptic membrane or structure, postsynaptic membrane or structure. From the presynaptic terminal, the neurotransmitter is released. It binds to the ion channels, which carry the receptor, which are the neurotransmitter receptors. And if they are ion channel linked receptors and they are fast-acting neurotransmitters, they will open sodium channels, and the sodium influx will depolarize the cell membrane, which therefore will form eventually an action potential, and that means that the postsynaptic cell gets activated. So this is how the excitatory synapse works. Now, it may happen, however, that the presynaptic membrane releases a different substance that has different receptors linked now to different ion channels. In this case, let's say chloride channels. And if these chloride ions will then after the neurotransmitter is bound open, then from the extracellular space, chloride ions will go into the cell, which, because of the negatively charged ions, will have an opposite effect on the cell membrane potential. Also, a similar effect occurs if another neurotransmitter is coming, which is linked to potassium channels. And as you know, in the extracellular space, the potassium concentration is much lower than intracellularly. After opening the potassium channel, efflux of potassium ions will take place. Therefore, again, the cell membrane potential will increase, hyperpolarization takes place, which finally will result in the decreasing electric activity of the cell, so an inhibition takes place. So this is how the fast-acting inhibitory neurotransmitters will increase the cell's membrane polarity, hyperpolarizes the cell. Because of this, the cell gets inhibited. So this is how the fast-acting excitatory and inhibitory neurotransmitters act. More details you will learn, of course, in pharmacology. You will learn more details about the receptors, about the kinetics, about the transmitters with many details, because if we would like to influence the neurons' activity, what we do is that we provide or we give medicines which interfere with this synaptic function by influencing neurotransmission. That's why that you later understand what the pharmacologists want to explain to you. We also have a topic about the neurotransmitters, how to classify them, and this is pretty much a table summarizing that field. So we have the acetylcholine, which is a specific, quite common neurotransmitter. We do not really classify it to any groups. We have neurotransmitters, the so-called amino acid derivatives, glutamate, aspartate, glycine, and gamma-aminobutyrate, or GABA, are also neurotransmitters. Some of those are excitatory, others are inhibitory, as I explained in the earlier slide. We also have chemical monoamines, which include the catecholamines. I point out the dopamine because the lack of dopamine causes, for example, a disease called Parkinson's disease, what you most probably heard about, but norepinephrine or epinephrine works as a neurotransmitter in the brain also. Histamine and serotonin are found mainly in the brainstem as neurons, but they innervate several brain areas, and they are involved in the mood control as well. And we have neuropeptides. The list is not complete. We have hundreds of neuropeptides already discovered. I just listed some of them. You don't have to know all of these, just at least some examples you could maybe learn. And other neurotransmitters like adenosine, anandamide, they belong to the purines and the cannabinoids. And finally, gas neurotransmitters, we also have, they easily diffuse through all cell membranes. Nitrogen monoxide, hydrogen sulfide, and carbon monoxide act also as neurotransmitters in the brain. How they are visible in the histological preparation is, of course, dependent on the technique what we use, but they show different kinds of synaptic vesicles. Either clear or so-called dense core vesicles are they related to. And I mentioned that some neurotransmitters are fast-acting, others are slow-acting. They have a longer effect. These are usually the G protein coupled receptors, and they have a complex intracellular signaling pathway. But you know about these intracellular signaling pathways more than I do from cell biology. And finally, I would like to show you something that is possibly interesting to you. So if you think about that we have around 100 billions of neurons, each of which has up to 100,000 synapses, and that number shows the number of total synapses in the brain. In each synapse, we have many neurotransmitters, and we may have several receptors. And it's of course also important at what time this signal comes, if the cell was at that moment activated or inhibited. It is also not unimportant where is the synapse located, because if here you have an inhibition on the dendritic shaft, but the dendritic spine is excited, the excitation signal cannot necessarily pass the inhibited surface of the cell membrane. This also interferes with the activity. And you shouldn't think that these dendritic spines are always there. They may develop and they may disappear within hours. And here I have some I think really nice images showing developing and disappearing dendrites, dendritic spines, which then explain why we can learn new things and why sometimes we keep forgetting some information. Because if the synaptic formation is supported, and we review, we recap or anatomy stuff, what we would like to remember for the exam, we will activate, we will enhance the synaptic plasticity, and new synapses will be formed, and they are supported and they live longer. Therefore, we can memorize things more efficiently for a longer period of time. Here I link the video in if you are interested. You can see a video how these dendritic spines can develop and how they, after a while, disappear. It's shown in a very interesting research. All right, this is what I had time for today. And with this slide, I would like to thank for your attention. And if you would have any questions, just let me know, and I would like to answer if try to answer if you have any questions.